welcome to the Independent Science News YouTube channel. So my name is Jonathan Latham and I'm the editor of Independent Science News. And what I want to do today is talk about whether GMO BT proteins are natural or not. So, you probably know that the GMO industry has basically two products, right? They have herbicide-resistant crops and they also have insect-resistant crops. And those, we're going to talk about the insect-resistant crops today. And those insect-resistant crops are basically producing BT proteins. So inside the crop is an insecticide. And so that means that you eat that insecticide when you eat the GMO corn or the GMO tortillas or sometimes the GMO oils, for instance, may have some of those proteins. But this is, largely an issue for whole ground up grains that people eat. And also, obviously livestock are eating these things too. Now, the whole sort of public defense of the biotech industry is that organic farmers also use BT crops or BT proteins. So the organic farmers use BT proteins. And these proteins are used by organic farmers as sprays. So basically what's being sprayed on an organic broccoli field, for example, will be bacteria which have inside them. So this is the Bacillus thuringiensis bacterium, and inside of that will be a crystal that is the sort of active ingredient of that sort of fermentation process. So it's true that organic farmers occasionally use these insecticides. But it's also, what is not true is that these proteins are the same as the ones that the GMO industry is using, right? And so we wrote a paper that came out this last September, which was to investigate the differences between the proteins that organic farmers use, and I'm not going to argue they use them on the same scale as the GMO farmers, because GMO farmers are basically, they're in every single crop all the time, produced preemptively. But essentially there are important differences between those. And I was aware of some of these differences, and we did some research. And so we published a paper in a journal called Biotechnology and Genetic Engineering Reviews. And this paper was written by myself and Madeline Love and Angelica Hillback. And Angelica Hillback is at the institute, the Swiss Institute. It's called ETH, and it has some incomprehensible name that I know the English translation, but that's not a lot of use to you. Anyway, so she works at this institute and she's a BT expert. So we brought her on board to kind of cover the basis of what we wanted to do. And what this paper does is lay out all the differences between the GMO proteins and the proteins used by organic farmers. And it turns out there's quite a list of those things, right? So in the first case, the proteins used by organic farmers are crystals. The proteins used in GMO crops are not crystals. The proteins used in GMO crops often are rearranged in various parts, so they're hybrids between different BT proteins. So they're also called cry proteins, by the way. So if I occasionally use the word cry, cry stands for crystal, but they're essentially the same thing. So the BT proteins produced in GMOs often also have mutations put into them. They also often are shortened, so they're smaller than the proteins used by organic farmers. They also can be modified inside the plant cell. So when you put a GMO transgene into a corn crop, for example, that corn crop then produces a protein which essentially is the answer is no one knows what kind of protein is actually made, right? Unless you go inside that crop and you actually measure it, you don't actually know what that protein looks like. But you can do analyses, which they do for risk assessments, and those analyses basically show that there's a whole diversity of proteins that are made in those GMO crops. And so they're producing proteins that are bigger than cry proteins ought to be. They're producing proteins that are smaller than cry proteins ought to be. And sometimes proteins of totally mysterious sizes. And so what this ends up being is a whole package of differences between GMO proteins and the proteins used by organic farmers. So the question then is, what is the biological significance? Like, is one lot of proteins more toxic than the other kind? And so that was where analysis went next. And the idea was, we looked at the theories of how these proteins are activated. Now, what's really interesting about these proteins is that they are the crystal form, which is used by the organic farmers, is not itself a toxin, right? In order to create a toxin, that crystal form has to be basically broken apart. It has to be broken apart from the original crystal. Then it has to be broken apart again physically. Then it has to be basically chewed up by a whole series of enzymes. It has to be shortened. And then you finally get to a toxic molecule, which is actually a lot smaller than the original protein. Now, what that means is, in practice, that sort of kind of counterintuitively, that smaller proteins are actually more dangerous, right, in many cases, than are the larger proteins. So the crystal itself is not a toxin. The partly degraded forms are not toxins. The even semi-degraded forms are not toxins until you get to the actual degraded form, which is actually a toxin. And basically what's happening is, the sort of analogy that you can use is that these are toxins that have safety catches on them. And a series of these safety catches has to be removed by the organism that ingests them in order for the protein to be activated. So what that means is that the smaller versions that are inside the GMO crop are actually the actual toxins, or are much more toxic than are the versions that are used by organic farmers, right? So if you ingested, for example, if a person ingested the organic spray, which is a crystal, you cannot digest that crystal, right, in the first place. But even if you could digest it, it's not certain that you would end up being able to cut it into the right sizes to be the actual disarmed or the activated toxin. So, but what is inside a GMO is often a pre-activated toxin. And so essentially, there are strong scientific reasons to be much more concerned about the proteins that are in GMO crops than the other proteins that are used by organic farmers. And so this is the paper that we wrote last September, which came out last September, and we explain in much more detail all the rationale of this and so on and so forth. But it has some important implications for, not only for people who are concerned about the toxicity of GMO, of these GMO crops, but also for the integrity of risk assessment, right? Why does it take us to come along and explain all this to the BT cry protein scientists? Why can they not come to these conclusions themselves, and why do regulators show no interest in these theses? And essentially, what I think is going on here is that the GMO BT crop community has basically a consensus which they built up, encouraged by the companies, the biotech companies, for a whole theory of GMO, of cry protein rather, toxicity, which is built around the question of whether you have receptors in your gut or not. And so they've built up this whole theory that you can't be harmed by a GMO BT crop unless you have the right receptors. What our interpretation of the data basically shows is that actually these receptors are a small and maybe even irrelevant part of the story of BT toxicity, right? But the whole receptor story, which has been kind of, is the quote unquote accepted set of theories around how cry toxicity works, is that story is essentially, it has very little support to it. And there is a whole part of GMO BT toxicity which is about the degradation of the protein until it's the right size and the right shape to actually make holes in gut membranes, because that is the toxicity that finally is the issue here, right? It's these proteins that make holes in lipid bilayer membranes. And lipid bilayer membranes are what separate your stomach contents from your internal organs, right? So these are proteins that make holes in lipid membranes. And so these are proteins that you should be concerned about as, as a personal human level in terms of toxicity, but also, the implications of all this not only are for human health of eating GMOs, they're also for livestock health, and they're also for non-target organisms, right? Any organism that eats GMO pollen, that eats GMO leaves, for example, or GMO roots, or tissues that fall into the ground, they all will basically have there'll be an unpredictability to the toxicity of all these tissues because of the unusual proteins that are being produced in these GMO plants. So there are all kinds of risk assessment implications of our thesis. And so this is the reason why people who work on cryotoxins and the companies obsess over whether target organisms have receptors, because that essentially takes them out of the space of having to discuss the actual changes in GMO crops that they're introducing into these plants. And so we have the classic example of where Monsanto has a patent on one of its GMO cryoproteins, and they call it a supertoxin. And then when they present it to the regulators, they say it's just the same protein that organic farmers are using, right? There's no reason to be concerned about this protein at all because it has a history, what they call a history of safe use. But that history of safe use, of course, presumes that the company hasn't come in and created mutations in it which enhance its toxicity. So it's a bit of a complicated story. And but if you go and read our paper, then this will be a good primer for that paper. But it's a very important paper, in my opinion, about GMO toxicity, because it provides a reason, right, for a rationale, a plausible biochemical rationale for how a person or an animal that eats GMO corn can actually be harmed by that GMO corn. So thanks very much, and I hope you'll join us next week for another video.